Okay, we start new point, monohybrid cross. So Mendel performed monohybrid cross, dihybrid cross, or even a trihybrid cross. So what is monohybrid cross? And what are the result of this cross? We learn in this session. In a monohybrid cross, Mendel select two pure homozygous plant. So the cross between monohybrid cross is a cross between two homozygous or pure parents by considering only one pair of contrasting character. So what is monohybrid cross? A cross between two homozygous or pure parents by considering only one pair of contrasting character. So in monohybrid cross, only one character or only one pair of contrasting character Mendel considered at one time. Okay, let us take example. In this cross, we cross the pure homozygous tau pea plant with the dark pea plant. So these are the parents. So these are pure homozygous. They have a genotype capital T. So these are the pure homozygous and small t. So this is the genotype of a parent. Okay, so these are the first parents or P1 parents, genotype of the parent, or this tall is the phenotype, dark is also the phenotype of a parent. Then gametes are formed. You have to make gametes. Homozygous individual, you know that they form the only one type of gamete. So capital T. This is also homozygous. They form the only one type of gamete. That is the small t. Then these gametes they fused together during fertilization to form F1 individual. This is capital T, small t. This generation is a F1 generation. He observed that in the first generation, all individuals are the tau. He known as a hybrid tau because it having one dominant and another recessive allele. So in the F1 generation, all individuals are formed. These are the tall individuals. Then he allowed for the selfing. F1 generation, that is capital T, small t, cross with capital T, small t. So that is selfing of F1 generation. And the gametes are formed. So it is heterozygous. They form the two different types of gametes. One is the capital T containing, another is a small t. Then this is capital T and a small t. So gametes are formed. These are the gametes. Okay. Then by using checkerboard or Punnett square checkerboard, you find out F2 offsprings. So Punnett checkerboard is given by the scientist Punnett. It is a box diagram in which, by using this diagram, you obtain the progeny of a F1 generation or zygotes of F1 generation. So in upper case, you write the male gametes. Suppose this is capital T, small t, and in lower case or in the left hand column, you write the female gametes. It is capital T, small t. So these male and female gametes, they fuse together to form the number of offsprings. So this T and this T, this gamete, male and female gamete, they fuse together. So capital T, they form the top individual. Then this capital T, dominant you write first, capital T, small t, this is heterozygous tall. Okay. Then capital T, small t, it is also heterozygous tall. And here small t, homozygous dwarf. And this capital T is a homozygous tall. Okay. So these four offsprings are formed in the F2 generation. So this is the F2 generation. If you observe these offsprings, in which this first offspring is tall, this is again tall, this individual also the tall. So we get 3 tall and 1 is the dwarf. 3 tall and 1 dwarf. So result, phenotypic ratio, you write the phenotypic ratio. Phenotypic ratio is equal to 3:1. That means 3 tall and 1 dwarf. 3 tall and 1 dwarf individuals are formed. And in phenotypic and in a genotypic ratio, see the genotype, capital T is homozygous tall. So this is only one individual. Then capital T, small t, these two, these are the heterozygous tall, and small t is a homozygous tall. So you write here. So genotypic ratio is 1:2, 2:1. So this is the result of monohybrid cross. Phenotypic ratio is 3:1 and genotypic ratio is 1:2, 2:1, in which 1 is capital T, which is a homozygous tall, capital T, small t, these are the two individuals, these are the heterozygous, and 1 is a dwarf, homozygous dwarf. So genotypic and phenotypic ratio obtained after this tall and dwarf cross, Mendel observed that in F1 generation, all individuals are obtained tall. So the tall character is expressed, or the dominant character is expressed. Tall is dominant character over the recessive. So the dominant characters are appear in a F1 generation. So this F1 hybrid, it shows the dominant character. So the characters appear in the F1 generation called as dominant character, and the character which is not expressed in the F1 generation is called as a recessive character. So by using this monohybrid cross, Mendel explained the law of dominancy. Law of dominancy states that, see, the first law of inheritancy, this is the law of dominancy. So, what is the law of dominancy? Mendel explained this. Characters appear in the F1 generation is called as a dominant character, or the allele they express in a heterozygous condition. These alleles are the dominant alleles. Suppose this is capital T and small t. This is heterozygous condition. In heterozygous condition, also a dominant traits they are expressed. So the law of dominance states that the characters appearing in F1 generation, these are the dominant character, and those are not appearing in F1, these are the recessive characters. Or even in heterozygous condition, alleles they are expressed, these are the dominant allele. So this is the law of dominancy. The first law of Mendel, that is law of dominancy, it is explained with help of monohybrid cross. This law, it is not universally accepted. Some exceptions are there. For example, incomplete dominancy and codominancy. We study further in this topic. So this law of dominancy is not universally accepted. Some exceptions are there. Then he gives second law with help of monohybrid cross. This is the law of segregation. Law of segregation. Okay, I think you remember, you understood what is law of dominancy. The characters appear in a F1 generation is called as dominant character. And what is law of segregation? See here, in F1 generation, whenever he crossed the pure tall plant with pure dwarf plant, in F1 generation, all individuals are formed, these are the tall. The recessive character, the dwarf character is not appear in a F1, but this character it is expressed in a F2 generation. So, during formation of gamete, when this F1 hybrid they form the gamete, these alleles capital T and small t, they are remain together, and during gamete formation, the factors or the alleles they get separate from each other and they enter into the different gametes. So there is formation of male gametes and a female gamete. So during formation of gametes, the factor or alleles they segregate or they separate from each other and they enter into the gamete. So the gametes are pure because gamete contain only one factor or only one allele of the pair. Factor, they are pure in nature. Why they are pure? Because factor, because gametes they are pure in nature because gamete contain only one factor or only one allele of the pair. So, the law of segregation is also known as the law of purity of gamete, purity of gametes. law of purity of gametes. Why the law of segregation is known as law of purity of gamete? Because during formation of gametes, the alleles they separate, or the factors they separate from each other and they enter into the gamete. Each gamete is a pure, they contain only one type of a gamete, either it is dominant or recessive. Therefore, it is known as a law of purity of gametes. So, these two laws, law of dominancy and law of segregation, they are explained by using monohybrid cross. In monohybrid cross, the phenotypic ratio is 3:1, in which 3 individuals are tall and 1 individual is tall. And the genotypic ratio is 1:2:1, in which 1 is pure or homozygous tall, 2 these are the hybrid talls, and 1 is a homozygous tall. So genotypic and phenotypic ratio, these are the different. So I think you understood what is monohybrid cross and what is the law of dominancy and law of segregation. Then after, in next point, that is a dihybrid cross, we learn about this.